In this video, we will show how to prepare and characterize biofunctionalized planar supported lipid bilayers. We will also describe in detail the architecture of a total internal reflection microscope with single molecule detection capabilities. We will measure the excitation power density, the fluidity and the protein density of the lipid bilayer. Let's begin with a short introduction to planar glass-supported lipid bilayers. When small unilaminar vesicles encounter a clean glass surface, they burst and spread out to form a contiguous lipid bilayer on a water cushion. We typically choose palmitoyl oleoyl phosphocholine, or POPC, as the predominant lipid because it gives rise to fluid lipid bilayers at room temperature and 37 degrees Celsius. For functionalization, we add nickel anti-ADOX, a synthetic lipid with a nickel chelating head group, which binds polyhistidine tag proteins, for example the co-stimulatory molecule B7-1, the adhesion molecule ICAM-1, and a fluorescently labeled peptide-loaded MHC molecule for T-cell recognition, as shown here. However, this bilayer system can in principle be employed to address entirely different processes in a variety of biological systems. Due to the nature of the planar system, fluorescence events can be monitored in total internal reflection mode, which cuts down cellular background substantially and is hence very well suited for single molecule fluorescence microscopy. For lipid preparation, one needs the lipids POPC and nickel anti-ADOX, chloroform and a clean round bottom flask. Dissolve the lipids in the desired ratio in chloroform and dry them under vacuum. As shown here, after high vacuum treatment overnight, the lipids form a film on the inside of the flask.